Can you see my screen?
Yes, sir.
No. Can you see my slide?
Yes, sir. You can go
My
to the
slide
full
is on
screen.
the screen now.
Yes, yes. On the screen. Yes. You can go for full screen.
I did full screen, but I don't know why it is not happening. Let me try again. Is it now? No?
No.
I don't know. Something is wrong with this setting. I think every time it happens like this. Okay.
This is fine. Okay. You can go in this mode also. Or you can close and again open the presentation.
Is it visible now?
Is visible.
Full screen is there?
No, it's not.
Can you see?
Normal mode.
I don't know what is wrong.
We'll start now. You can also go in this mode.
One.
Yes.
Okay. That's the best I can do right now.
Yes, it's okay. Students, we are entering into the second lecture for the day. We have with us Dr. Sandeep Goyal, scientist chef at the National Institute of Animal Biotechnology, Hyderabad. Dr. Sandeep Goyal is a veterinarian with a PhD in reproductive biology from Kyoto University, Japan. He spent 20 years at the Center for Cellular and Molecular Biology, CCMA, Hyderabad, as a scientist and a team leader. There, he researched reproductive biotechnology of domestic and wild animals with a specific emphasis on stem cell biology, animal transgenics, testis biology, spermatogenesis, and cryobiology. He has authored more than 50 scientific papers, book chapters, and research articles. Dr. Goel was a visiting faculty of Georgetown Medical School, USA, and Kyoto University, Japan. Dr. Goel is currently working at the National Institute of Animal Biotechnology, Hyderabad, developing mesenchymal stem cell cryobanking for allogenic and autologous transplantation to aid in the widespread clinical application in livestock and companion animals. So, with this short introduction, I invite Professor Dr. Goel to deliver his talk on role of biotechnology in animal production. Doctor, the floor is yours.
Okay, thank you, Dr. Murugan, and I thank BIIS for giving me this opportunity to connect with the students through you. And I'm sorry if I'm not able to project my slide properly, but believe me, I know there is some technical glitch from my end, which I am not able to, but anyways, I'll do justice to, I hope you can see my slides, right? I mean, that should be okay, right, Dr. Murugan? Yeah, it's okay now?
Yes, it's okay.
Yeah, okay, fine. So,
But now,
yeah, so today I'm going to talk about role of biotechnology in animal production. And, I actually belong to the institute called National Institute of Animal Biotechnology, which is located at Hyderabad. And this has a wonderful campus and also, runs a PhD program and master course dissertation program for people who are interested in animal biotechnology. So I would, encourage you to apply for these programs and keep your eyes on the website of NIB, where you can find these advertisement and you can pursue your dream in these areas. Okay. All right. So coming to biotechnology, the definition of biotechnology are several, and I find this particular biotechnology definition to be more appropriate. And over the period of time, I somehow identify this with this definition more and more. It's a set of techniques by which living organisms are modified for the benefit of humans and other animals. Okay, so this is what I can think. Actually, there are several definitions of biotechnology, and biotechnology is something which is very ancient. It started with brewing of wine and making of bread. And so many. There was a genetic selection of animals throughout these. And we have the kind of banana that we see today is actually coming from the wild type of banana, which was full of seed. And it is just because of the selection process of biotechnology that we are able to eat a banana which is seedless. Okay, and so is the case with the kind of animal that we move around. The kind of animal that we have around us has been extensively selected through genetic selection and phenotypic selection to make them valuable for us for the milk production and for the meat production and for several other reasons. So if you see biotechnology, it is actually an interdisciplinary science which not only consists of biological science together, such as genetics, microbiology, and food science, or the biochemistry and chemistry, but also several engineering science, such as biochemical engineering, chemical engineering, mechanical engineering, and food technology engineering, and they all come together to form this science known as the biotechnology. So basically, if you split this biotechnology into two parts, it's biology and the combination of technology. Okay. So emphasis in animal biotechnology currently are three. So that is, the first one is the breed management, where I'm going to talk more about in more detail. Then is the health management and feed and fodder management. So that is what is the emphasis of animal biotechnology with respect to livestock production. Although there are other animals, such as you have pet and companion animals such as dog and horses, where the emphasis is different on their taking care of their. So it's more or less different. But we are talking in more emphasis is in this particular lecture. We're going to talk more about livestock industry. So biotechnology and animal production, I'm going to cover up three basic biotechnology area. That is reproductive biotechnology, transgenic technology, and genome edit technology, which I'm going to talk about a little bit more in detail. So, please be around. And I think that some of these terminologies can be really challenging for younger students but I will try my level best to explain it as I go through. So reproductive biotechnology is a very, very important area of biotechnology in animal reproduction, and it has really revolutionized the white revolution is because of one of the technologies that I'm going to talk about today. So if you see reproductive biotechnology in animal, we have several technologies, and most of them is here. The artificial insemination, one of the major, as I told you, is one of the major technologies that revolutionized the white revolution in India by increasing the milk production. And then we have in vitro fertilization, we have sperm sorting of sperm, and reproductive cloning and embryo transfer technology. So I'll talk about one by one in a little bit brief so that you can have an understanding. So artificial insemination, or also known as AI, please don't confuse with artificial intelligence. Many of the time people use this, but AI was used almost 50 years. This technology is in place now, and it's been extensively used in animal reproduction. So if this is a news clipping from a 2000 newspaper where 70 cows, it shows that 70 cows are impregnated every hour through artificial insemination in Madhya Pradesh. Okay, so MP is one of the most extensively using this technology, followed by Telangana, in which the Hyderabad is located. So this news is kind of in 2022, 2 years before. So now the situation might have been a little more different, but it appears to me that Madhya Pradesh still leads in utilizing this technology for reproduction in animal in livestock production. So artificial insemination by definition is the deliberate introduction of sperm into the female uterus or cervix for the purpose of achieving a pregnancy through in vivo fertilization by means other than sexual intercourse. So artificial insemination means that you deliberately introduce the sperm into the female uterus or cervix. I'll talk about this more in detail as we go forward. So as you know, that the animal, usually most animals are cyclic, that is they come in estrus, or estrus is also known as the sexual receptivity, where they are sexually receptive for reproduction or breeding. So in case of cow, they come in estrus every 21 days, where there is the change in the hormone profile of estrogen and progesterone. You can see here that this is the time when estrus behavior takes place somewhere, and very high level of estrogen is there. And at the end of the estrus period, there is an ovulation. So this period lasts for 12 to 18 hours in case of cow. And this is a small window where the animal has to be inseminated for fertilization to take place. So the recurrent set of physiological and behavioral change that takes place from one period of estrus to another is known as the estrus cycle. Okay, so this is what happens in case of cow in 21 days, and this is the time window where the insemination has to be done or the breeding has to be done. Okay, and what happens at this particular window, there is a release of an oocyte from the ovary, which travels all the way down to from the fallopian tube or the oviduct. You can see here. And in the oviduct, the sperm travels all the way from the vagina to cervix through the uterus and through the oviduct, and the fertilization takes place in the oviduct. And once the fertilization takes place, the oocyte is activated to undergo cell division, where it undergoes 2-cell, 4-cell, eventually 8, 16, and eventually forming a morula and a blastocyst. And after the blastocyst hatches out from the zona pellucida and it is implants itself in the uterus, and therefore setting in the pregnancy. Okay, so this is generally about what happens in general. Most mammals, this kind of an event takes place. So the objective of artificial insemination is that it gives you a very important tool of genetic improvement. Okay, so you can actually breed a cow I mean, maybe she may be genetically superior with a very genetic superior male sperm. Okay, which is very important for getting a very good quality offspring, which can produce better milk for the dairy industry. So genetic improvement is one of the objectives. Superior offspring, as I told you, better disease control, because since the male does not come in contact with the female, many of the disease can be controlled from through this technique. The possibility of increasing fertility, you can have male fertilizing many females. So in a lifetime, if a male can breed around 100 females or 200 females, by using this technique, he can breed maybe 500,000 females, So this is the advantage of this technology. And it also decreases the breeding expense, because one cement straw can only cost you like 50 to 100 rupees or 150 rupees, depending on the quality of sire or the male. So that decreases the breeding expense. The advantage of AI is that, as I told you, it is genetic improvement, widespread use and availability of genetic superior sires. One bull can breed 500,000 cows in a lifetime using this technology. And even after death, semen can be used and can be kept in liquid nitrogen for a long term, frozen for a long time. Older semen so far has been used after 40 to 45 years old, so that has been kept in for almost 50 years and kept in the liquid nitrogen can be used. It is a rapid proof of male superiority. So since one male can semen can be used for breeding several number of females, so it gives you a very quick rapid proof of male superiority by progeny testing these offspring. And then and as I told you, that natural mating can only produce 100 offspring, like the max. But here you can see that 50,000, 500,000 cows can be inseminated, and maybe thousands of offspring can be produced. As I told you, it is availability of sire is is everywhere now. You can ship the semen straw from one country to another or one state to another. Danger of male is removed, disease is better control, and crossbreeding can take place, and improves the record keeping of the farm. And it is very economical to use, because since one during one ejaculation, a male produce billions of sperm, and these sperms can be extended and frozen into a small. So for a fertilization, for an artificial insemination to take place, maybe 5 to 6 million sperm is good enough. Okay, so if from one ejaculate, around 300 to 400 semen straw can be produced. Okay, so that is a very good way that one ejaculation can be used for extending the semen. And cost of maintaining the sire is also reduced in the case of farm. Also, it has some disadvantages, that there has to be a trained inseminator for doing this technique, because it requires a little technical skill, and the heat detection or estrus detection has to be good. Also, the freezing of semen is only limited to certain species. Still now we are struggling to freeze other animals sperm or semen, but so far in case of bull, it is a very good technique. And sometimes what happens, that a poor male can also move into the semen can also move into the system, so that has to be taken care of. And maintaining the long term storage can also be a technical issue for several areas, because availability of liquid nitrogen is important. So I'm just putting up some of the picture of the exotic breed, cross breeding, and native breeds here. So you can see here, these are some of the indigenous breed of our India, Sahiwal, Gir, and then we have these exotic cattle, and there has been a cross breeding program which is going on in India for several decades, that we are trying to cross breed the exotic cattle with the indigenous cattle for enhancing the breed production, and artificial insemination has been very instrumental in doing so. So I will just go into the brief of the steps of artificial insemination. First is the semen collection. Then we evaluate the semen. Then we extend or dilute the semen. As I told you, that one ejaculate has several billion sperm, at least 2 billion sperm, and we can make, and for fertilization to take place, we need just some millions sperm. So we can dilute this semen to make several number of straws, and then freezing then these extended semen, and then insemination of the cow in estrus. Okay, so just to tell you in brief, we take the elite bull or the best bull, we collect the semen from the elite bull by artificial vagina, and this collected sperm is then, the semen is then extended, diluted, and then it's then frozen, and this filled up into this liquid night, this straws. This is known as the this French straw, which contains 0.5 ml or 0.25 ml of semen, extended semen. And once you fill up these extended semen into these straws, you freeze them in liquid nitrogen, and then you can keep them in liquid nitrogen for as long as you want, because in liquid nitrogen, which is minus 194 degrees centigrade, you can keep it for long term. And then you can store it into these liquid nitrogen, and when required, and you can see that these are some of these liquid nitrogen which contain the semen from different bulls which has been frozen and can be utilized in the future for artificial insemination. These liquid nitrogen container are shipped to different places in our country. For example, if the Telangana has a very good quality bull, they freeze the semen of this bull and transport it to Madhya Pradesh or any other place like Jammu and Kashmir, and so many places it can go. So you can see that the best bull don't have to move from Telangana for the breeding. It can, the liquid nitrogen container can be moved, which contain the semen straw of this bull. So once we need to do insemination in the cattle, this semen straws are removed from the liquid nitrogen container, and then they are warmed up in 37 degrees water bath. You can see here, and one of the end is cut. The crimped end, and that is loaded into this AI gun or artificial insemination gun, which consists of this steel and then a sheet over it. So when you put this semen straw in front of these AI gun, and then cover it with a plastic sheet, and then it is ready for artificial insemination. So once you have the AI gun ready, then you have the cow which is in estrus or in heat. You pass the semen straw, the AI gun through the vulva into the vagina and into the cervix, and then you using another hand into the rectum, you guide this AI gun through the cervix. You can see here, this is a cervix here, and in the interior part of the cervix, the semen is deposited in case of cow, and this is a very important place to deposit the semen. If you deposit it elsewhere in middle of the cervix or in the vagina, the conception rate is reduced. So the exact site of insemination is very important. And once the insemination is done, you remove the AI gun and let the animal go and wait for it to see whether it has the conception is set in. And then a trained veterinarian can tell within 45 days through rectal palpation if the pregnancy has been set in or not. If it is not set in 21 days, the animal will come back in estrus again. Another technology that I'm going to talk about is in vitro fertilization, also known as the test tube babies in case of humans. So here, the IVF is the process of creating embryos from oocyte, that is unfertilized egg, by fertilizing them with semen in a petri dish, that is in vivo and in vitro, sorry. And the oocytes are first collected from the ovaries of the donor by ultrasound guided follicular aspiration, and they are matured in petri dish and fertilized 20 to 24 hours later. And then oocyte is then developed into incubator for several days, at which point the resulting viable embryos are transfers into the recipient animals. Okay, so this is how it's a flow chart. You have an elite cow, you do a ovum pickup, and you collect the oocyte, and using the elite cell sire semen, you in vitro fertilize it. And this in vitro fertilized embryo can be cultured to produce blastocyst, or can be frozen for later transfer, or they can be directly transferred into the recipient animal to produce the live offspring. This is how it is done, the ultrasound guided. So we insert the ultrasound probe into the vagina, and with one arm in the rectum, we guide it, and then through this needle, we aspirate the oocyte from the antral follicle. And once the aspirated oocyte is kept in the petri dish, they are matured, and then they are subjected to the capacitated spermatozoa. And you can see post-fertilization, these oocyte gets fertilized, and they produce the embryos which are cultured to appropriate stage, and then they are transferred into the surrogate mother for the implantation. So using this technology, we can, like for example, using ovum pickup, you can in vitro mature and fertilize. You can also do a genetic selection of embryos, where you can take a biopsy of an embryo, a small piece of tissue cell is taken from the embryo, and then using a genome selection, or using a DNA chip or genome chip, you can actually select the embryo based on the genotype, and then you can use the best embryo for embryo transfer for producing a genetically superior offspring. So I must tell you that NIAB was produced a DNA cattle genome chip called as Indigao, which is India's first cattle genome chip for the conservation of pure variety of indigenous cattle of the breed, which was released for the public by His Excellency Minister of Science and Technology, Dr. Jitendra Singh, last year. Okay, now coming to the sex sorting of sperm, which is another technology which is used in reproductive biotechnology. As you all know, that the male, the semen or the spermatozoa decides the sex of the fetus or the offspring, because the sperm are either X bearing or the Y bearing. So if the oocyte is fertilized with X-bearing sperm, it becomes a female, and if it's fertilized with a Y-bearing sperm, it becomes a male. So fluorescence-assisted cell sorting, or FACS, is used for sorting of spermatozoa based on the DNA content of the sperm, as we know, as I'll show you, that the X-bearing sperm and the Y-bearing sperm differ in the quantity of DNA content. So X-bearing sperm has slightly more DNA content than the Y-bearing sperm, and so you can see here that the testes produces X and Y-bearing sperm, and then we add vital dye to it. Vital dye is a dye which binds to these cells, but they do not actually kills this the spermatozoa, and only the live sperm can actually take up this dye. And then you can see here that the X-bearing sperm takes more dye than the Y-bearing sperm, and using a laser sorting system where it is detected by a computer, you can see here when the sperm is passed through a very narrow field, and then they are individually passes through a laser detector and a computer laser point, and then a computer detector. A magnetic field is created, and this computer detecting system actually codes the sperm through a fine covering of for a charge on it. The X-bearing sperm are given a positive charge, and a Y-bearing sperm are given a negative charge. And using a magnetic field, these sperms are deflected into, sorting into Y and the X-bearing sperm, and the dead sperm, which do not actually takes any dye, can also be sorted out into a separate area. So you can see here that using this technology, we can separate X and Y-bearing sperm in this case. And you can know, if we inseminate a cattle with only a female X-bearing sperm, you will only get a female. And it is very important for a dairy industry to have female calves coming out, because it's the milk from the female animals, not from the male animals. So therefore, this technology is really important for increasing the milk production in case of dairy industry. So coming to another technology, reproductive cloning, which was a very, very major hype in the 90s, 97, 96, where the Dolly the sheep was produced for the first time. However, it has lost its charm over the period of time because of its technical difficulty. So, in India, this is a photograph from ICR Center of Central Institute of Research in Buffalo, where they have been using this technology for producing the cloned buffalo animals. You can see here. So the cloning, by definition, is the process of producing similar population of genetically identical individual through asexual mean of reproduction with the use of sperm. Okay, so let me talk to you something more about this in detail. So advantage of cloning is that it restores endangered species and conserve prized animal. It allows for editing of gene in the farm animal. I'll talk about gene editing. And it is an alternative method of generating transgenic animal also, for making bioreactors or farming. Okay, so basically the very simple technique of cloning that we have seen in case of humans also is the identical clones that we have. Basically what happens that during the fertilized egg, the two cells stage embryo, and you can separate these blastomeres into single cells, and if one of the blastomeres, so since they're coming from a similar genetical identical, fertilized embryo, so they can be actually a clone of each other. Now, this is how it is done in case of in case artificially it is done. So under the microscope, we can actually hold the egg and remove one blastomere, and then finally, the blastomere is transplanted or transferred into a empty zona pellucida to produce a two cell embryo splitted clone. Okay. Then another method that can be used is the blastocyst bisection. So at the blastocyst stage, you can cut open the zona pellucida and bisect the embryo under the microscope with a knife in equal halves, and then you can put these equal halves into zona pellucida, and then they can develop into each can develop into an identical clone of each other. So nuclear transfer, as I told you, this is a technique where the nucleus from a cell is transferred into an enucleated oocyte, and then these somatics, nucleus actually decide the genetical composition of the embryo. Therefore, it is known as a nuclear transfer. So genetically, this embryo that is formed from the enucleate outside actually has a genetic gene copy of these cells, which is coming from a. This is what it is called as a nuclear transfer. So there are two kind of nuclear transfer. One is the embryonic cell nuclear transfer, where the donor nucleus comes from an embryo. That is what is a very common method of doing, because the embryonic cells are very easy to reprogram as compared to the somatic cells. Therefore, embryonic cell nuclear transfer are rather more successful than the somatic cell nuclear transfer. So in this case, you can collect the blastomere nucleus, and then you enucleate the oocyte, and then you can put the blastomere nucleus or the and then you can develop this into. So each of these nucleus can produce one individual, and these are therefore genetically identical to each other, since they are coming from a common oocyte, common embryo. Okay. So the another technology which is known as somatic cell nuclear transfer, through which the dolly, the sheep, was produced, is somatic cell is an any cell of the living organ other than the reproductive cell. So they are terminally differentiated cells. So what happened that from somatic cell, you can collect the nucleus from there, and then you can enucleate the oocyte. And if you replace the somatic cell nuclei into the oocyte, this is all known as the somatic cell nuclear transfer. This technique has been used extensively in the past decade, but has been very inefficient because of several reasons. And epigenetic reprogramming is one of the major constraints that has discouraged the use of this technique in the future. Another technique that terminology that people often use is therapeutic cloning, but in case of humans, since this technology is banned because of ethical reason, however, they have been using this technique for using therapeutic reasons. So what they do is they take the skin cells. For example, if I suffer from any disease and I need my specific stem cell, so what they can do is they can take the oocyte and replace it with my skin cell and produce a blastocyst, and from the blastocyst you can derive a pluripotent stem cell. So if technically speaking, this particular stem cell belongs to me because the genetic material is coming from my body, so therefore I can use this for my own therapy. So that is known as therapeutic cloning. Okay, coming to another technology which is very important in case of animal reproduction is embryo transfer technology. So in this technology, what happens that in this, the embryo is collected from a donor female. So in this case, if you remember the artificial insemination, genetical superior male produces more offspring in case of artificial insemination, but in case of embryo transfer, genetic superior female produces more offspring than they can usually do. Okay. So in the case of embryo transfer is a process by which an embryo is collected from a donor female and then transferred into the recipient female, where the embryo completes its development. By the use of embryo transfer, a genetic superior female produces more offspring than she could by natural reproduction. So in general, a cow can produce in a lifetime at the maximum maybe 5 to 10 calves. But using this technology, she can produce a calf in one cycle itself, around 10 to 15 calves in one cycle itself. I'll show you. So basically what this technique is, you take a donor, which is a genetically superior, you superovulate it by using hormonal injection, where you superovulate the animal to produce more number of oocyte. Then you do an artificial insemination, you fertilize all of these oocytes to produce embryos. And these embryos can be transferred into the recipient, which are estrous synchronized to produce the offspring of these genetically superior donor. Okay. So this is a picture of a donor here, which has produced, see here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 offspring in one cycle. So this is how the embryo transfer technology works. Okay. So advantage is that it increases the number of offspring sired from a superior female. It results in faster genetic progress, obtained females offspring from old or injured animals. So in this case, if an animal is injured or old, you can still get a calf from that animal. It increases farm income through embryo sales. So you don't have to transfer all of these embryo. These embryo can be frozen or cryopreserved and can be sold to another farm for the and can be shipped to any other place for importation or exportation of the embryos. Okay. So the step of AI, as I'm going to go into brief detail, you take a superior female animal, you superovulate with hormone injection, and after that you do an artificial insemination 5 days after the superovulation, and you collect the embryos non-surgically by by draining by flushing the embryos through the uterus using this particular catheter system, and then when you isolate the embryo, you evaluate the embryo based on their morphology, and then classify, and these embryos can be actually cultured for some time or stored in the liquid nitrogen, or they can also be transferred into a surrogate mother to produce a live offspring. Okay. So in general, this is what superovulated ovary looks like. You can see here that during a normal estrous cycle, only one antral follicle can be seen. But in case of superovulation, you can see here that 1, 2, 3, 4, 5, around 8 to 10, oocytes are produced from one ovary. So the left and the right ovary can produce maximum up till 20 to 25 oocytes, and they can be, fertilized during artificial insemination. And this is how the draining of the collection of embryo is done. You insert a catheter into the uterus, and then you flush the uterus with the collection media, and then you collect it from the other side, and you can collect these embryos. You can see these embryos look like in a different, so the good quality embryo looks like this, expanded blastocysts or blastocysts, so they're graded, and then these are transferred into the recipient female, which are estrous synchronized through transvaginal method. So result of ETT, you can see here that this mother has produced maybe 10 to 12 calves in one cycle of her superovulation. So this is really amazing to increase the productivity of a female, genetically superior female. Coming to the next part of my lecture, I'm going to talk a little bit about transgenic animals. The transgenic animal is an animal in which the carries a transgene, that is a foreign gene that has been deliberately inserted into its genome. The transgene is constructed through a recombinant DNA technology, which consists of a promoter region, a protein coding region, and polyadylation signal. So depending on the kind of interest that you want to produce in the transgenic animal, you can use this gene. Nowadays, people are using it for producing, pharmaceutical protein in the milk of the in the milk of the animal. So this is how it is done. You micro-inject the DNA into the fertilized embryo. You can see here, and then, or even you can use somatic cell nuclear transfer, and then you transfer these injected embryo into the surrogate mother to produce a transgenic animal. Okay. So the transgenic animal are currently being used for human health, for the production of pharmaceuticals, production of xenografts, especially pigs, been used for producing xenografts or the organs for human use as a disease model, production of dietary supplement of functional food, and also answer environmental problems. Okay. So in conventional method for recombinant protein production, you have the fermenter or the tissue culture technique. But however, now the transgenic technology, we have been able to produce transgenic sheep, goat, and transgenic cattle and transgenic pigs. So this is now a reality. We can see them now in the industry. So they are actually used for making pharmaceutical or the gene farming for efficient scale up, very correct glycosylation and post-translational modification of the protein can take place. It reduces enormously the cost of production, and the transgenic founder can be rapidly propagated, and the stability of the transgene is very, very stable. Therefore, what happens that these are some of the products which has been produced in the milk mammary gland of the transgenic animal, like for example recombinant human antithrombin 3, which has already been approved for marketing, and then you have several other like esterase inhibitor, albumin, fibrogen, collagen, lactoferrin, alpha-1 antitrypsin, malaria vaccine, and some of these monoclonal antibodies has been produced in the mammary gland of the transgenic animal. So if you see the cost of goods here, so if you see here that the transgenic animal, the cost of good in dollars per gram, you can see here that when compared to cell culture, you can see here that only $6 per gram if you are able to produce 300 kg of this per year. So you can see the cost of production of these transgenic proteins in the animal milk is far cost effective than the cell culture technique. So some of these goat has been used for producing pharmaceutical protein like spider milk, lysozyme, factor 9, lactoferrin, antithrombin 3, and there have been extensively used in the industry. Another example which has been used for making transgenic salmon fish. Here you can see here, this is a transgenic fish which expresses growth hormone, and this you can see has the growth of this animal. This fish is tremendously very much as compared to its non-transgenic age-matched fish. So you can see here that it has, and this has actually been marketed. These fishes are available on the counter for consumption. Coming to my last part of the talk, I'm going to talk about a little bit about genome editing, which is the new techniques has been introduced. So what is genome editing? Basically, genome editing or genome engineering is a type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the genome of a live offspring. In 2018, the common method for such editing used was known as engineered nuclease or molecular scissors. Okay, so this is a central dogma. We know that DNA produces RNA and RNA produce polyproteins. So the genome editing actually works at the DNA level, where it modifies the DNA of an animal to produce a genetic modified animal. So this is the very common method used maybe decades back, where when we did not have these new genome engineering tools. So homologous recombination was a most common method of using genome editing tool, where we use the homologous arm, the 5' and 3' region, and put a drug resistant gene in between, and then we introduce this targeting vector into the cell, and our gene of interest, when they align and undergoes a homologous recombination, replace the gene A promoter region or the exonic region with the drug resistant gene, and this is how the chromosome or the gene was disrupted in a chromosome. This was a very conventional method, and it was very inefficient. Only 1 to 3% success rate was there. And then came these new genome editing tool known as the zinc finger nuclease, Talon, and CRISPR-Cas9, which has revolutionized the way we are now, editing the genome of any organism. So this three technology, and CRISPR-Cas9 specifically, is much more specific and very efficient, can cause double stranded break in the genome, which can be either done can be replaced either by a donor DNA for targeted gene replacement, or it can actually cause an indel targeted mutagenesis by non-homologous end joining to produce the mutant offspring. So these are the methods which is now currently being used extensively for the genome editing. Okay, so as I told you on my earlier slide, that in vitro fertilization in animal is a very common is a method for making a donor female genetically superior female propagation. So here, if you can use in vitro fertilization, you can actually introduce the genome editing here in this in vitro fertilized oocyte, and you can these embryos can then be screened, and then the genetically modified animal can be then transferred into a surrogate mother to produce a offspring which is a genetically modified and genetically superior. Okay. Similarly, genome editing tool can also be used for modification of male germline. So if you're donor male, you can collect the spermatogonium stem cells, and then you culture these stem cells, and you can do a genetic modification using the CRISPR-Cas9 or Talon or zinc finger nuclease, whatever method is convenient for you. And then you can check for the validate the gene editing in spermatogonium stem cell, and then transfer the spermatogonium stem cell back into a recipient animal, and these genetically modified SSCs can produce genetically modified spermatozoa, which can be used for artificial insemination or in vitro fertilization. And then you can see a genetic offspring can be produced using this technology. Another method which I was talking about, somatic cell nuclear transfer. So genome editing technology has actually increased the through 25 to 50%. If you see here, earlier transgenic swine or transgenic animal was produced by injecting the DNA into the to the nucleus of the embryo, and you can see here the efficiency was very low. And then came somatic cell nuclear transfer, where they were using gene targeting method. And then you can see here the homologous recombination method only gave less than 2 in 10 to the power 4 targeting, efficiency. But using this modern gene editing technique like zinc finger nuclease, TANN, and CRISPR, you can see this efficiency has increased to 25 to 50%, which has really important in the case of animal biotechnology, where we have been able to do these gene editing with so much high efficiency. Okay, so coming to the summary of my talk, biotechnology has revolutionized the animal production by enhancing the reproductive and genetic capabilities of the animals. Several biotechnological innovations are in pipeline that are going to further enhance the production capability of animals. There's a need for fresh talents like you to continue this innovation in this field. But however, ethical issue pertaining to these technologies should always be kept in mind and should be always done within the purview of the ethical, guidelines of the country. Okay, thank you so much for your attention, and I'm now open for questions from the audience.
Thank you so much, Doctor. The session is open for discussion. If you have any questions, you can ask Dr. Goyal.
So there is a question, I can read it here. As you have talked about freezing in many part of your project presentation, what is the future of cryogenics preservation in terms of storing human eggs and sperm? And can the storage of neurons is also possible in this manner? Okay. So yes, there is a lot of work going on, and people are freezing human eggs and sperm. So it's not nothing new about it. We have a lot of IVF clinics and many places where women now feels that they are professionally very busy, so they don't want to have children right now. So they are actually freezing their oocyte that can be used in the future for their conception. Okay, so even sperm and human sperm is being frozen as we talk about every day. And there has been a lot of research going on for the efficient freezing, so that when they are thawed or they are unfrozen, their survivability is very high. Okay. And talking about the neurons, see, so neurons are terminally differentiated cells, as you all know. So freezing of terminally differentiated cells has always been little bit difficult. However, sperm is also a terminally differentiated cells, but since they are in millions and millions, their survivability, even it is 70%, is pretty good enough, right? But for the storage of neurons, now they are developing new techniques, especially they are using nanotechnology to combine with freezing these neurons so that they're efficiently frozen and then they can survive the freezing process. So new cryoprotectants are coming up, new techniques of freezing such as vitrification, where high amount of cryoprotectant are being used to develop these technologies.
Next question. Results. no more questions, then I would like to thank Dr. Goel for joining us today. Thank you so much.
Thank you so much, Dr. Murugan, and thank you everyone for being such a wonderful audience and attending this lecture. Thank you so much.
Thank you.
Okay, bye. Thank you.